Alright, welcome to the third part of the urinary chapter. So far, we've had discussions on an overview of the urinary system. We've talked about the anatomy of the kidneys. We've had sort of an overview of renal physiology. And we've gone into the details on glomerular filtration, as well as tubular reabsorption and secretion. But now we're going to go into some additional renal physiology, things like the regulation of urine concentration and what regulates the volume. We're going to talk more specifically about urine and how it clears the kidneys, and how it is transported, stored, and eliminated from the human body. So let's go ahead and get started. Now, as you guys have learned so far, about 85% of water reabsorption that happens in the kidney is obligatory. Water is obliged to follow via osmosis, right? That last 15% is going to be facultative, okay? And that is going to be adjusted by hormones to meet the body's needs and maintain that homeostatic balance. Facultative water reabsorption is what's going to really determine the final concentration of urine and the volume of the urine. And in this part, we're going to talk about how the kidneys adjust for facultative water reabsorption and produce either dilute or concentrated urine, as well as like the hormones that play a part in this process. So if you trace the path of this filtrate, you start at the glomerular capsule. And as this filtrate exits the blood and enters that first part of the renal tubule, it is generally isoosmotic, basically equally osmotic as plasma, at about 300 milliosmoles. Do remember that in the nephron loop, this osmolarity begins to change because of the differing permeabilities of both the ascending and descending limbs, right? So the first part is the thin descending limb, and it is permeable to water, but not solutes. And so water is going to flow out of the filtrate into the interstitial fluid through osmosis, but very, very few solutes are going to follow. And so this makes that filtrate more concentrated. And by the time it reaches the bottom of the loop, it's about 900 milliosmoles, about 300 times more concentrated than it was when it began. And the exact concentration actually depends on the length of the nephron loop. A longer loop contains more concentrated filtrate at the bottom. So those juxtaglomerular nephrons are going to concentrate the fluid more than the cortical nephrons. As the filtrate begins to enter the thick ascending limb, sodium and other ions are going to be pumped out of the filtrate because now it's permeable. However, this part of the limb is virtually impermeable to water, so water doesn't follow these solutes. And the concentration of the filtrate decreases actually as it moves up the ascending limb. And by the time it exits the thick ascending limb into the distal tubule, its osmolarity is generally less than that of the filtrate at the same level in the thin descending limb. So it's somewhere in between now. In the early distal tubule, ions are going to keep leaving while water stays behind, and it decreases even further to about 100 milliosmoles. And this is going to be 3 times less concentrated than it was when it began. Once the filtrate enters that late distal tubule and the collection system, the principal cells are going to begin this facultative water reabsorption, that last 15%. And so this concentration is going to vary by the amount of water reabsorbed. If less water is absorbed, concentration of the filtrate stays low. The end result is the production of dilute urine, or urine with a concentration of less than 300 milliosmoles. However, if more water is reabsorbed, concentration of the filtrate increases as it passes through the late distal tubule, and so it's a more concentrated urine, meaning that it has a concentration of greater than 300 milliosmoles. And we're going to talk about the means by which both dilute urine and concentrated urine are produced here in just a second. All right, so the thin descending limb is going to be permeable to one of these, right? Which is it? It's water. Remember, the descending limb is made up of that simple squamous epithelium that only allows water but not solutes to pass through. So when the extracellular fluid of the body contains too much water, it's going to produce dilute urine. The kidneys are going to produce dilute urine. And so that means that the filtrate entering this late distal tubule is already less concentrated than the surrounding interstitial fluid, right? So for this reason, the kidneys simply just turn off facultative water reabsorption, and this produces dilute urine. Physiologically, this is done through a reduction in ADH release. The loss of antidiuretic hormone renders those principal cells of the late distal tubule and the collecting system essentially impermeable to water. And do note that the principal cells continue to reabsorb sodium and chloride ions from the filtrate, so the number of solutes in the filtrate is going to lower while the amount of water remains the same. And so this is going to cause the concentration of the filtrate to progressively decrease as it passes through the system. It's going to become more watery. And when there is an excessive amount of water in the extracellular fluid, this osmolarity of the urine can fall as low as 50 milliosmoles, which is only 1/6 of the osmolarity of plasma. And typically, the volume of urine produced also is going to increase when this urine is very dilute. Normally, the volume of urine is about 1.8 liters. However, when ADH secretion is low like this, the urine volume can increase dramatically. And do note that urine volume is also increased by other factors like fluid intake, general health, diet, and blood pressure. And if a patient is taking diuretic drugs, then that can act on various parts of the renal tubule to block reabsorption of water or solutes and promote diuresis. So there are a number of factors there, but the ones we're focused on right now are just the natural extracellular fluid being too low in concentration. Here's a pretty good diagram showing what we're talking about. So you see the filtrate is going to leave this the glomerulus at 300 milliosmoles, and as it passes through the proximal tubule, it's going to lose a little bit of water obligatorily, but mostly it's going to be losing mostly it's going to be losing water. And then as it progresses into the descending limb, you're going to see that it becomes much, much more concentrated. As remember, these limbs here are only permeable to water. And so through this whole region, it's only losing water. And it's becoming more and more concentrated, right? And you see it gets to 900 milliosmoles down in the bottom of the loop. However, once we finally get to the ascending portion, the thick ascending portion, it can finally become permeable to solutes and not just water. And actually, it only retains water because it's impermeable normally to to passive diffusion. And so it begins losing sodium chloride, right? It loses sodium chloride, it loses any other ions or solutes. And so as it passes through, up into the distal tubule here, you see it's lost. It's lost almost 800 units of osmolarity here by the time it gets to the distal tubule. And by the time it leaves the distal tubule, it's gone way over that. And so it just continues losing solutes as they're reabsorbed, right? And then we get to this ultra low concentration of urine production, which is mostly water, relatively few solutes. It's dilute urine. Now, on the other hand, the kidneys are really effective at conserving water. They can produce really, really concentrated urine, up to 1,200 milliosmoles. And this is the human body. The Australian desert hopping mouse is actually capable of producing urine at a concentration of 10,000 milliosmoles. So these kidneys are really, really effective. And this concentrated urine is really going to result from basically turning on facultative water reabsorption, right? And so this is going to be in the principal cells of the late distal tubule and the collecting system. And this is going to be stimulated mostly by ADH. However, it does require more. If you guys remember, water reabsorption only happens by osmosis, and osmosis only occurs if an osmotic gradient is able to drive it. So that means that facultative water reabsorption will only take place if the interstitial fluid, the extracellular fluid, is more concentrated than the filtrate. And that makes sense. There's less water in the interstitial fluid, more water in the filtrate. Water is going to flow from high to low. And the interstitial fluid within this renal cortex has about the same osmolarity normally as all the other interstitial fluid, about 300 milliosmoles. By the time the filtrate enters the late distal tubule, it has an osmolarity of about 100 milliosmoles. And so this interstitial fluid around the late distal tubule and the collecting duct is going to be more concentrated than the filtrate. And this is going to create a very small but significant gradient that drives water reabsorption. However, by the time the filtrate enters the medullary collecting duct, enough water has typically been reabsorbed that its osmolarity is about equal to the interstitial fluid, and the gradient is going to disappear. If the osmolarity of the interstitial fluid as a whole, renal medulla, remained with 300 milliosmoles, there's going to be no further osmosis, and water reabsorption is going to be finished. And so to continue reabsorption and production of concentrated urine, the nephrons have to work to create this osmotic gradient within the renal medulla. And so this is going to be called the medullary osmotic gradient. This medullary osmotic gradient is going to start with the medullary interstitial fluid. It's going to start with the medullary interstitial fluid at a concentration of between 300 and 400 milliosmoles at the cortical medullary border. And this is going to increase to 1200 milliosmoles, 4 times more concentrated than plasma, at the deepest regions of the medulla. And this is going to be established and maintained by a countercurrent mechanism. This is a mechanism that involves fluids flowing in opposite directions that exchange materials or heat. And this is going to have 3 components. First, you have the countercurrent multiplier system that's found within the nephron loops of these juxtamedullary nephrons. Next, you have the recycling of urea in the medullary collecting ducts. Okay? And third, you're going to have a countercurrent exchanger located in the vasa recta. All of these work together to make that concentrated urine. So if we talk about the countercurrent multiplier, do remember that we're talking about 2 different types of nephrons, right? The cortical and the juxtamedullary. We've been talking up to this point about the physiology of both types of nephrons, but now we're going to talk specifically about juxtamedullary nephrons with these long nephron loops descending deep into the renal medulla. With these, you're going to find the countercurrent multiplier helping to establish this medullary osmotic gradient. And in this system, countercurrent is going to refer to the fact that the filtrate in the 2 limbs flows in opposite directions. The filtrate in the descending limb flows toward the renal pelvis, and the filtrate in the ascending limb flows back up toward the renal cortex. This is going to be called a multiplier because the 2 limbs of the loop are close enough together that a small osmotic difference at the top is multiplied as we drive further down the loop. And so it basically works like this. Sodium potassium chloride symporters are going to pump sodium chloride from the cells of this thick ascending limb into the interstitial fluid, right? So they're pumping it out of the filtrate. And so now water can't follow it, and the accumulation of the sodium chloride in the interstitial fluid increases its molarity. This concentrated interstitial fluid is going to create an osmotic gradient that is going to draw water from the filtrate in the thin descending limb now into the interstitial fluid. And then, if you guys remember, the nephron loop's thin descending limb is going to be impermeable to solutes, right? So it remains in the filtrate as the water leaves. And so for this reason, the filtrate becomes progressively more concentrated as it reaches the bottom of the nephron loop. More solutes, less water. And by the time it reaches the thick ascending limb, the filtrate has a very high sodium chloride concentration. And this is key because the symporters in the thick ascending limb function in proportion to this sodium chloride concentration. The higher the sodium chloride in the filtrate, the more that is pumped back into the interstitial fluid. And so this is how this countercurrent multiplier is able to achieve such a high sodium chloride concentration in the interstitial fluid in the deepest part of the medulla. Do note that this is, as it reaches this concentration of about 1200 milliosmoles, there's going to be a significantly higher value of the nephrons, which reaches about 300 milliosmoles. And then as the filtrate moves up through the ascending limb, more sodium chloride is pumped out, and the sodium chloride concentration in the filtrate is going to finally begin to decrease. When this occurs, pumps will progressively drive out less sodium chloride into the interstitial fluid, so the sodium chloride concentration will progressively decrease as it goes higher up into the medulla. These symporters are going to begin this step here in this figure you're going to see in a second. And do note that all of these steps typically are occurring at the same time, but in this figure you're about to see, they're separated for simplicity. So this is going to be how this countercurrent works, okay? So first we start here. We start at the end, this thick ascending limb. As fluid travels up, it's going to pump all that sodium chloride out, right? And so this is going to be actively transported out into this interstitial fluid. And because these two, the descending limb and the ascending limb, are so close, they actually affect each other. So as this sodium chloride is pumped out in the ascending limb, it's going to be pumped into that interstitial fluid, and the high sodium chloride concentration in here now draws out extra water in this thin descending limb. So extra water is going to be pumped out here, and it's going to really, really concentrate the filtrate. And you see here, it's going to get super, super concentrated. The sodium chloride concentration gets ultra high down here at the bottom of the loop. And eventually, we get back up into the ascending limb, and because it's so highly concentrated, it's going to allow this sodium chloride reabsorption. As it reaches this thick ascending limb, it's going to reach this gradient of hot, ultra high to normal, and it's going to pump itself out from high to low. And so it's a cycle that perpetuates itself. So I hope that's a good way to understand this sort of countercurrent multiplier that occurs in these nephrons. Now, salt, sodium chloride, isn't the only solute in the interstitial fluid, right? Another solute helps establish this medullary gradient, and it's urea. As urea diffuses into the interstitial fluid from the medullary collecting duct through a process called urea recycling. And here's how it works. As water is reabsorbed from the filtrate, it's going to leave behind the urea, right? And the urea is going to become more concentrated in this fluid. In the medullary collecting ducts and in the papillary ducts, urea is going to follow a concentration gradient from high to low and passively diffuse out of the filtrate and into the interstitial fluid. This is going to contribute to the existing gradient, right? Where some of this urea is going to re-enter the thin descending limb and rejoin the filtrate. And the urea remains there until it reaches the medullary collecting duct again, where it again diffuses back into the interstitial fluid. Do note that this urea is diffusing out of the medullary duct, and it only consists of a small amount of the total urea. Much of it remains in the filtrate and is going to be excreted in the urine. So there's a very small amount that's recycled, but it is significant in contributing to the medullary osmotic system here. Now, normally, when solutes reabsorb into the interstitial fluid, they go into the capillaries and are carried away, right? This presents a problem in the renal medulla because they can't produce a concentrated urine without that gradient in the interstitial fluid. So, this means that reabsorbed sodium and chloride ions can't immediately be swept away by the blood. However, this renal medulla has to have its blood vessels, as its cells need oxygen and nutrients. And so for this reason, we have those special capillaries around these juxtamedullary nephrons to create this, to maintain this medullary osmotic gradient. The vasa recta, remember, we talked about this sort of lateral-looking structure. And just like the limbs of the nephron loop, the vasa recta descend into the medulla, and then following a hairpin turn, ascend to the renal cortex. Do notice, and when I show you this photo, they are arranged so that their flow is opposite to that of the nephron loop. The descending vasa recta parallel to the ascending limb of the nephron loop, and the ascending vasa recta parallel to the descending limb. Okay? And this arrangement is a countercurrent flow acting as a countercurrent exchanger that can supply blood to the renal medulla cells with while maintaining this medullary osmotic gradient. And so here's how you can see this countercurrent exchanger working with the descending limb of the vasa recta and the ascending limb of the vasa recta. So first you start with the descending limb. You see coming off of the efferent arteriole here, we follow it, and it eventually becomes the descending limb of the vasa recta. And the descending vasa recta is going to lose water and gain sodium chloride as it, encounters this medullary interstitial fluid. This is going to continue as it progresses down and around the loop. And then eventually we get into the ascending vasa recta, which exchanges its water for sodium chloride. And it basically just works to even itself out. And you see as the vasa recta enters this countercurrent, it's at 300, and as it leaves, it's at 300. So it has a net change of 0, but it works to synergize with the this nephron loop, maintaining this medullary gradient. The big picture of what's happening here in the vasa recta is the descending vasa recta picks up salt and loses water, right? Then the ascending vasa recta exchanges these two substances by picking up water and losing salt. In the end, these cells of the renal medulla obtain oxygen and nutrients, but we maintain the medullary osmotic gradient that is established by the countercurrent mechanism and urea cycling. This is a super simplified model that I just showed you, as there are numerous vasa recta and nephrons actually that almost touch each other in the kidney, but it's going to be like, think of it just like a simplified artistic rendition, but it gets the idea across. Now, which of the ascending limb of the nephron loop, right? This is going to do what? Ascending limb of the nephron loop does what? It reabsorbs sodium and chloride. Remember, ascending limb is permeable to solutes, but not water. Descending limb does which? Water only. Remember that. So it's inverted. Countercurrent multiplication is best described as which of these? It involves filtrate flowing in opposite directions through those limbs of the loop, right? As the filtrate flows down through the ascending limb and up through the down through the descending limb, up through the ascending limb. And so they're going to counter-currently exchange. Blood descending into the medulla in the vasa recta, blood descending into the medulla of the vasa recta, so this is the descending vasa recta, does which of these? It gains sodium chloride by losing water. Remember that. So the countercurrent system is going to be really complicated, but let's take time to summarize this function. Keep in mind that the entire function of this system is to conserve water when needed, right? Water can be reabsorbed only if antidiuretic hormone is present, right? And this osmotic gradient is there to drive osmosis. This medullary osmotic gradient doesn't exist on its own, and so the kidneys have to create it through the following mechanisms. The countercurrent multiplier of the thick ascending limb establishes the medullary interstitial current gradient by pumping salt into the interstitial fluid, right? More salt in the interstitial fluid is going to drive water toward it. Continued solute reabsorption, including urea recycling from the filtrate in the medullary collecting duct, is going to add to the gradient. More solute in the interstitial fluid drives water reabsorption. The countercurrent exchanger of the vasa recta allows perfusion of the inner medulla while maintaining medullary interstitial gradient. As the descending limb loses salt, it's going to drive water reabsorption, right? All of this drives water reabsorption. And so all of these combine to produce concentrated urine. So let's look at this as these steps are combined. When filtrate enters the cortical collecting duct, there is no osmotic gradient, right? So no water is reabsorbed. And this means that filtrate, entering this medulla has the same concentration as the interstitial fluid. So there's no osmotic gradient present. But in the presence of antidiuretic hormone, the concentrated medullary interstitial fluid is going to create a gradient for water reabsorption. And this interstitial fluid in this case is going to be more concentrated than the filtrate. And so the filtrate passes deeper into the renal medulla through the medullary collecting duct. And if antidiuretic hormone is present, water is going to be drawn back into the interstitial fluid via osmosis, thanks to those aquaporins that are created. And water is reabsorbed until the filtrate and the interstitial fluid are going to become isoosmotic. Deeper into the medulla, this interstitial fluid becomes more concentrated, so water reabsorption continues from the medullary collecting duct. And it continues because the interstitial fluid has become progressively more concentrated in the deep renal medulla. So more concentrated interstitial fluid, more intense water reabsorption. And eventually, you get concentrated urine that's produced. This produces a urine with a concentration of up to, like I said, 1,200 milliosmoles. And we cannot make more concentrated urine because after this point, there's no longer a gradient to drive osmosis. It exits into the renal pelvis and is transported into the ureters, and we'll talk about that next. Now, let's say there's too much ADH secreted, right? This is going to be called syndrome of inappropriate ADH secretion, or SIADH. And this is characterized by too much antidiuretic hormone. And it has multiple causes, including ADH secreting tumors. Like certain lung tumors are actually especially prone to this. But regardless of the cause, the result is the same. Excessive fluid retention that leads to a decreased plasma osmolarity and a high urine osmolarity. So this is just one of those conditions that can really result in increased urine concentration. So urine volume is actually not going to be influenced by one of these. Which one is it? Gender. There's no influence on gender, by gender on urine production volume. Now, as filtrate passes through the nephron, volume tends to decrease the most in the proximal tubule, right? It's going to lose the most amount of water in those regions. Maximal urine concentration is limited by, which of these limits the maximum urine concentration? The medullary interstitial fluid concentration, right? And so that's why if we make the medullary interstitial fluid super concentrated, it's going to drive that massive amount of water reabsorption. This one photo is an entire section in your book, okay? I want you to really look at this book because it breaks the entire physiology of this nephron into 6 basic steps. And I've already talked about these, but I'll just go over them briefly. First, you have glomerular filtration, and then you have what regulates the glomerular filtration rate, right? Then you have reabsorption and secretion in the proximal tubule. Once you leave the proximal tubule, you have the countercurrent multiplication and exchange in the nephron loop. After that, you have reabsorption and secretion in the distal tubule. And finally, you have the production of either dilute or concentrated urine as regulated by the medullary collecting duct. So all of these working together, this is the sort of big picture here of what we're looking at as far as renal physiology. And if you at any point do not understand anything about this, please let me know. I am here to answer questions. Changing gears here. So far we've talked about how blood is filtered and how filtrate is modified, right? Now we're going to look at the final product that exits the papillary ducts and is excreted. This is the urine. And once it leaves the papillary ducts, it is officially urine. We're going to look at the normal components of urine and any abnormalities that can signify problems. We're going to also look at another way in which the health of the kidneys can be assessed through a value known as renal clearance. So urine is going to be the fluid that remains, right, after tubular reabsorption and secretion have taken place. Normally, what does it contain? Water, sodium, potassium, chloride, hydrogen, phosphates, sulfates, metabolic wastes like urea, creatinine, ammonia, things like that. It also can contain like bicarbonate ions or calcium or magnesium ions. And there are certain abnormalities in the urine, conditions that can cause abnormalities. And these, so an analysis of the urine or a urinalysis, big shocker on how that name came up with, or how they came up with that name. This urinalysis can be a valuable tool in the diagnosis of a disease. And the variables examined in a urinalysis are color, translucency, odor, pH, specific gravity, things like that. Color is typically urine is typically colored by a yellow pigment called urochrome, which is a breakdown of hemoglobin. The more concentrated the urine, the less water is present. So the same amount of urochrome makes urine dark yellow. So once, if a lot of water is being reabsorbed, you're going to have that dark yellow urine. The color of urine may also be altered by certain foods, vitamins, drugs, food dyes, or the presence of blood. I know that certain antibiotics can turn urine almost neon orange. And that's just a little bit of useless trivia for you. You also have translucency. Translucency, regardless of color, the urine should always be translucent. You should always be able to see light through it. If the urine is cloudy, that typically indicates an infection, but it could also indicate that the urine has a large quantity of protein in it. Recently voided urine should have a mild odor, but if it's allowed to sit out, bacteria are going to metabolize the urine to produce ammonia, giving it a much stronger odor. And the odor of urine may also be altered if certain disease like diabetes mellitus or infection, or by eating certain foods like asparagus. that old adage, oh, I shouldn't have had asparagus. That's actually true. It affects the odor of the urine. Now, the pH of urine, normally around 6.0, slightly acidic, but it has a range from 4.5 to 8. And the reason that it's a minimum of 4.5 is that the hydrogen ion transport pumps in the distal tubule cannot pump against a higher hydrogen ion gradient than 4.5. Specific gravity compares the amount of solutes in a solution to the amount in, let's say, deionized water. Deionized water has no solutes, and so thus is assigned a specific gravity of 1.0. Urine has solutes, so its specific gravity will be higher than that, and that can range from 1.001 to 1.035. And 1.001 is very dilute, 1.035 is very concentrated. And there are a number of other properties like the presence and relative levels of solutes, and they're analyzed with urinalysis test strips. Test strips are dipped in the urine, and their chemical indicators change color in the presence of specific substances, commonly testing for blood, protein, leukocytes, glucose, or more. And if the kidneys are functioning properly, these substances should not be present in significant amounts. Now, although urinalysis provides valuable clues about renal function, a more complete assessment is provided by measuring the rate at which kidneys remove a substance from the blood. This is called renal clearance. And the renal clearance of the chemical is then used to estimate glomerular filtration rate. Renal clearance and the GFR both are measured in the same unit, milliliters of plasma per minute. So for a substance to provide an accurate measure of renal clearance and GFR, the substance should be completely filtered and neither reabsorbed nor secreted. Substances secreted by renal tubules have a renal clearance greater than their GFR, whereas those that are reabsorbed have a renal clearance less than their GFR. And so to measure renal clearance, we have a limited number of substances from which to choose. Two commonly used substances are creatinine and insulin. So creatinine is going to be this waste product of muscle metabolism and other cells. Nearly all creatinine that's produced is going to be excreted by the urine. But when the kidneys are impaired, this level of creatinine tends to rise. Generally, a plasma creatinine of 1.2 milligrams per deciliter, is considered abnormal. So anything above 1.2 mils per deci is abnormal, but it does tend to vary with age, sex, and body mass. Excretion of creatinine can be used to estimate GFR by comparing the amount of creatinine excreted in the urine to the amount of concentration of plasma the plasma concentration of creatinine. So, and the main difficulty of using creatinine as an indicator is that between 15 and 50% arrive there via secretion and not filtration. So a patient with a very low GFR may still have a very normal result, right? It may bypass this filtration and just be secreted straight in. A more accurate assessment is used with insulin. I'm sorry, inulin, not insulin, inulin. Inulin, not to be confused with insulin, is a complex carbohydrate found in plants like garlic and artichokes, and it is filtered by the glomerulus, but it is neither reabsorbed nor secreted, so this GFR may be accurately measured by injecting inulin and comparing its excretion in the urine to its plasma concentration. Now, let's talk about getting rid of the urine, all right? The urinary tract is going to consist of two ureters, the urinary bladder and the urethra. And we're going to talk about their anatomy and their histology here and the differences between males and females. So first we start with the ureters, and the ureters are going to transport urine from the kidneys to the urinary bladder. They're generally about 25 centimeters long with about 3 millimeters in diameter. in an adult, and they begin roughly at the level of the second lumbar, traveling behind the peritoneum, emptying into the urinary bladder, right? Just like any hollow organ, they have a multi-layered wall, and their three layers, from superficial to deep, are the adventitia, the muscularis, and the mucosa. This adventitia is going to be a fibrous connective tissue that basically supports the ureters. The muscularis is going to be a smooth muscle-containing middle layer, and that's like the smooth muscle of the alimentary canal. This contracts rhythmically via peristalsis. Waves of peristalsis course through the muscularis as often as 5 times a minute, depending on the rate of urine production, and it all works to propel urine toward the urinary bladder. Deepmost, you have the mucosa, this membrane consisting of transitional epithelium and an underlying basal lamina. As you guys learned in chapter 4 when we talked about histology, a transitional epithelium is going to be stratified with cells that can change from having a domed shape to being squamous. It allows them to expand and recoil. So the ureters are going to drain into the posterior inferior urinary bladder. In this region, there is a mechanism that prevents urine from backflowing. As each ureter passes along, it travels obliquely through a tunnel in the bladder wall. And eventually, as urine collects in the bladder, pressure is going to rise and compress that tunnel, pinching it closed, preventing backflow. The bladder itself is a hollow, distensible organ sitting on the floor of the pelvic cavity, suspended by a fold of the parietal peritoneum. It collapses when empty, but when it's distended, it almost becomes pear-shaped, and it can hold between 700 and 800 milliliters of urine. Although in females, it doesn't actually expand to full capacity because of the uterus's position. Like the ureters, the walls have 3 tissue layers. From superficial to deep, they are the adventitia, the detrusor muscle, and the mucosa. And do note that the floor of the urinary bladder contains a small triangular area called a trigone. This trigone is going to lack rugae, and it appears smooth because the mucosa is tightly bound to the underlying muscularis. And the 2 posterior corners of the trigone are formed by the 2 ureteral orifices that have those mucosal flaps that act as valves, right? And this apex of the trigone is going to be formed by the opening to the urethra, this internal urethral orifice. Now, like I said, the 3 tissue layers are going to be the adventitia, the detrusor, and the mucosa, from superficial to deep. The adventitia is going to be composed of areolar connective tissue, and on the superior surface, there's going to be an additional serosa. That's the fold of the parietal peritoneum. You have the middle tissue layer composed of smooth muscle called the detrusor muscle. And these muscle fibers are arranged into inner longitudinal, middle circular, and outer longitudinal layers. And so this muscle forms a circular band around the opening of the urethra called the internal urethral sphincter. And eventually you get down into the innermost layer, the mucosa, and it is composed of that transitional epithelium with an underlying basal lamina. This has a mucous membrane that produces mucus, protecting the bladder epithelium from urine. And when the bladder is not full, folds of mucosa called rugae are visible. Let's say what would happen if this urine wasn't present. We'll talk about that with interstitial cystitis. But let's look at this actual photograph and the illustration and this histograph of the urinary system here. You start with the ureters up here, and you have a cross section with the histology of the ureter showing the different layers. You have the mucosa. In deepmost, you have the lumen. Remember, it's hollow, so it has that hollow space. Then you have the mucosa, the muscularis, and the adventitia on the outside. So once you leave the ureters through these urethral openings here, you see they have those sort of flaps that'll fill back and prevent backflow. But the bladder itself, you see, has all of those various muscular layers crisscrossing. You have longitudinal, circular, longitudinal, and that makes up this detrusor muscle layer. At the very top, you have the parietal peritoneum resting on it. All of it surrounded by this adventitia on the outside. And eventually you get this internal urethral sphincter, this muscle band here that holds the bladder shut. And you do see this sort of trigon made between the urethral opening and the urethral openings. That's just the sort of quote-unquote triangle. The other thing you would need to notice out of this is the rugae. In this case, it's an artificially inflated bladder, but you can still see these folds here of the internal surface of the bladder. And then we have a histograph here of the bladder itself. Again, we start with lumen, and then you have the mucosa made up of what kind of cells? Transitional epithelium. And here you have the detrusor muscle layer. So it's relatively simple, but just make sure you guys understand these layers and the histology, because it's all test-worthy material. Now, as I said, inadequate mucus production results in a condition called interstitial cystitis. This allows acids and other toxic substances to damage the underlying mucosa, right? IC is characterized by frequent urination and pelvic pain coming from mucosal ulcerations, not lacerations, ulcerations. And the underlying cause of interstitial cystitis is typically unknown and unfortunately responds very poorly to treatment. Over the long term, IC tends to lead to bladder scarring and to contraction. It's a really unfortunate condition, and I hopefully, no one suffers from that in this class. Something I forgot to mention is that the position of the bladder does differ a little bit in males and females. In males, it is anterior to the rectum. In females, it is anterior to the vagina and inferior to the uterus. We'll see a photo here in just a minute showing the comparison. But the last part is the urethra. And this is the terminal portion of the urinary tract, draining urine out from the bladder to the environment, the exterior of the body, right? Like the rest of the urinary tract, it has an outer adventitia, a middle muscularis, and an inner mucosa. And it begins in the urinary bladder, right, which is surrounded by this internal urethral sphincter. This sphincter remains closed unless urine is voluntarily being eliminated or involuntarily being eliminated. Now, there is an external urethral sphincter. This is formed from the levator ani muscle, okay, also sometimes called the urogenital diaphragm, made out of the muscular floor of the pelvic cavity, right? So this is going to be the sphincter that is composed of skeletal muscle and is totally voluntarily controlled. At the external orifice of the urethra, there is no sphincter. The levator ani creating this external urethral sphincter is the last stopgap for both males and females. And although the urethra does structurally differ, it does differ structurally, but it also functionally differs a little bit as well. Female urethras are shorter, about 4 centimeters, opening at the external urethral orifice between the vagina and the clitoris, and it serves exclusively as a passage for urine. The male urethra is longer, whatever the length of the penis is, in addition to the length of the females, right? So the male urethra consists of 3 regions. There's the prostatic urethra, the membranous urethra, and the spongy urethra. And the prostatic urethra is going to be what exits the urinary bladder, right? And it passes through the prostate gland. And the prostate gland sits inferior to the bladder. The membranous urethra is going to be the shortest segment, and this is what passes through the levator ani muscle. And then you have the longest segment. This is the spongy urethra, passing through the penis to the external urethral orifice. And it is called spongy urethra because it passes through the erectile body of the penis called the corpus spongiosum. And the location of the male urethra allows it to serve a dual purpose. It transports both urine and semen. We'll talk about that when we get to the reproductive chapter. But here you can see the different regions. You see females only have a urethra. It exits the bladder and exits the body. The urine exits the body. Males have these. Males have this prostate gland, and as the urethra passes through the prostate, it is the prostatic urethra. And then you have the very short membranous urethra as it passes through the levator ani muscle. Eventually, all you have is the spongy urethra, and eventually you get to the external urethral orifice, eliminating this waste from the body. And right about where the prostatic urethra ends is where this dual function begins. You can see right here the seminiferous tubules begin to empty into this urethra, and that's where the dual purpose actually begins. So micturition is the actual term for urination. This is the discharge of urine from the urinary bladder to the outside of the body. It is activated by a reflex called the micturition reflex, mediated, of course, by the parasympathetic nervous system. This reflex arc is carried out by three components. There are stretch receptors in the urinary bladder walls. There are sensory afferent nerve fibers that convey this information to the sacral portion of the spinal cord, specifically S2 and S3. And eventually you have parasympathetic efferent fibers that travel to the detrusor muscle and the internal urethral sphincter. So micturition basically occurs when urine fills the bladder and stretches its walls. This micturition reflex is going to occur. In infants and young children, this involuntary reflex is the primary mechanism through which they urinate. And it proceeds as follows. Stretch receptors send a signal, right, through sensory afferent fibers to the sacral portion of the spinal cord. And then it's processed in the spinal cord, and parasympathetic efferent fibers stimulate the detrusor muscle to contract and the internal urethral sphincter to relax. This causes micturition. And the micturition center in the pons of the central nervous system, given time and training, makes that a voluntary process. And you can see this reflex here. As urine fills the bladder, it stretches those walls. The walls send those signals, hey, we're being stretched, to the spinal cord. And the spinal cord sends those interneurons to the micturition center in the pons. Pons, if it's developed enough, it will send signals back to the efferent signals to both the detrusor muscle and the levator ani muscle and the internal sphincter, urethral sphincter, causing them to either relax or constrict, depending on which muscle it is, and it will just squeeze that urine out of the bladder. This photo puts everything all together: urine formation, urine storage, and elimination. So now that you've examined all these processes, this is one final big picture view so that you're able to trace the formation, the storage, and elimination of urine. Overall, you see blood flowing into the glomerulus. It gets filtered. We go through the whole filtration and reabsorption and secretion process. And then you get down to urine drainage, where that newly formed urine drains out of this papillary duct into a calyx, a minor calyx, from a minor calyx into a major calyx, into the renal pelvis, from the renal pelvis into a ureter, from a ureter into the bladder, from the bladder through the urethra into the environment. That's the general traced out pathway feel free to look at this is another good photo to look at in your book to sort of review. And if you have any questions at any time, do not hesitate to ask. Now, let's do a little bit of test yourself before we end. Active transport in the thick ascending loop, limb of the loop. Active transport in the thick ascending limb of the nephron loop is going to be driven by sodium potassium 2 chloride symporters, right? They're moving things in the same direction. So which of these is the reason that the countercurrent multiplier is considered to be a positive feedback? It's because the events in each limb amplify each other, right? The descending limb pulls out water into the interstitial fluid. The water that's there pulls out solutes from the ascending limb, right? So they sort of just feed into each other. And as more solutes are in the interstitial fluid, that pulls more water out from the descending limb. So it just sort of piles onto each other. The vasa recta does what of these? Contains blood flowing in the opposite direction from the filtrate in the loop. Remember, blood flows down the ascending and then up the descending. So this is the sort of stuff you just covered in this section. We talked about reabsorption and secretion, production of dilute urine, production of concentrated urine, urine composition and analysis, urinary tract anatomy, which is super basic, and the process of micturition. This is it for this chapter. Chapter 24 is in the bag. If you guys have any questions, you know what my email is. Do not hesitate to contact me. I will see you guys in the next one.